Okay, so if you've made it this far, great job. And we finally get to start putting some of this information that we've been learning for the previous 8 chapters to some applicable use. So the stuff we're going to learn from here on out will have direct clinical application as well as just life application in general. If we've gone in order, then we're going to be bringing stuff up from all of the previous chapters. If we've gone out of order, then just keep some of the stuff in mind that we're going to discuss when we get to some of the earlier chapters later. So this chapter is essentially focused on how do we kill these microbes, primarily focusing on pathogenic microbes. So controlling these microorganisms is obviously something we need to be concerned about because they cause diseases, right? And what we're going to be focusing on in this chapter is controlling the microbes outside of the body, meaning we're not talking about medications and whatnot. We are talking about cleaners and disinfectants and uh, ways to control the populations of microbes um, on surfaces and instruments and things like that. So there's 4 possible outcomes to controlling them. Sterilization, and we're going to talk about all of these in detail. Disinfection, decontamination, sometimes called sanitization or sanitation potentially, or antisepsis. So one thing that we need to be very, very aware of is not all microbes are quote unquote germs. Okay, germs, as we know that term to be defined in our common language, is a bad thing that causes some sort of a disease or infection or disorder or whatever you want to call it. So this table right here in your book just kind of lays out the basics of all of these different means of controlling these microbes. So let's talk about some kind of key features on this really quick. So sterilization here, one thing to be aware of is this term all. Sterilization kills everything, including viruses, including prions, everything. There's nothing that survives sterilization. And so when we use these words, you need to understand the definition so that you can use them properly. So for example, if you use, Pine Sol or something to clean a surface, you are not sterilizing it. You're disinfecting it. So you wouldn't want to use the term sterilization, and so you just got to be careful not to mix these up. Another couple things. This sterilization is all or none. There's no such thing as being partly sterile or slightly sterile. It's either sterile or it's not. It's black and white. And so this is used for inanimate objects like surgical instruments and things like that. So we don't sterilize body parts. That would be impossible. If you sterilize a body part, the only way to do that is via cremation. So the body part does not survive. And the way we do this, a couple of ways, and we'll talk about this in detail, is either heat, and we use an autoclave, or a chemical sterilant. Okay, and sterilants by definition kill everything, right? And so they're able to destroy the spores, which these other methods down here do not, at least not completely, because the spores are much more resistant to these methods than the vegetative forms. So what are we exactly trying to control and why? So the things that we're primarily trying to kill these microorganisms for is because they either cause infection or they cause spoilage of our food. And one thing that's important to understand is these things, these microbes rarely live as alone as a single species. There's almost always several species or genera or both that we're trying to control all in the same process, either on the same piece of equipment or the same food or something along those lines. So the goal of sterilization is to kill everything, which includes these spores. And if we're killing a spore, we're killing the vegetative cells as well, because there's nothing that we know of that can kill a spore without killing the vegetative cell. So in other words, if you're killing a spore, you're killing everything. Now this chart right here shows the relative resistance of different species and genera and even kingdoms. So we start down here with the least resistant, which are these enveloped viruses. And I don't want you to necessarily memorize this, but at least be aware that there is a continuum all the way up to things that are more and more and more resistant. So these trophozoites right here, for example, are more resistant than an enveloped virus. Staph and Pseudomonas happen to be more resistant than gram-positive bacteria. Non-enveloped viruses are fairly unresistant. And we get all the way up into here, this end, that are very resistant are these bacterial endospores and then these prions, which remember were proteins that caused those spongiform encephalopathies like mad cow disease. And they are very difficult to kill. So this chart is trying to show how much more endospores are resistant than the vegetative forms, okay? And so over here on this side is the method. So for example, if we use heat, it takes 120 degrees Celsius for a period of time to kill an endospore, where it only takes 80 degrees Celsius for the same period of time to kill a vegetative form of the same bacterium. And so it is 1.53 or just 1.5 times more resistant, the endospore is, than the vegetative forms. And then if we use radiation in the form of X-ray, it takes 4,000 grays compared to 1,000 grays. So it's actually, the endospore is 43 times more resistant to radiation than the vegetative form. Sterilizing gases like ethylene oxide is 1.73 times more. And if we're using a sterilant chemical like glutaraldehyde, a 2% solution of that, it is the endospores are 183 times more resistant than the vegetative form. So if we're using this to sterilize what would take 10 minutes to get rid of the vegetative forms, we'll have to leave the sterilizing liquid on for 3 hours to kill the endospore. So there's two different things that we use to, or the, I guess two different methods, I guess, that we use to control microbes, either agents or processes. So processes are steps that we take. For example, if we say we sterilize something or we disinfect something, that's a process. Agents are chemicals, such as a bactericide. And we may use a bactericide, so we would use an agent as part of a process, potentially. But if we use a bactericide, but a bactericide is a chemical that destroys bacteria, but does not kill the endospore. A sporicide is a type of bactericide. So sporicide kills bacteria, but it also kills the endospore. So a sporicide, even though I've got it listed under bactericide because it is a bactericide, it's really kind of its own category. A fungicide is a chemical that obviously kills fungi and includes the fungal spores. A viricide obviously is something that kills um, viruses, or kill is probably the wrong word to use because remember viruses aren't considered to be alive, so it inactivates them. A germicide or a microbicide is a broad category that just kills microorganisms. And it kills all microorganisms, good, bad, whatever. It's a broad category and won't necessarily tell you if it kills fungi or endospores or anything. We just know it kills something. So there's this term sepsis. This is not an agent. Sepsis is a term that we use that defines the growth of microorganisms in the blood or other tissues. So if you are septic, that means you have bacteria living inside of you, floating around in your blood is generally the term that that's used for, or the condition that that term is used for. So asepsis then is any practice that prevents the entry of the infection into the body, okay? So in other words, it just prevents infection. So aseptic techniques are the practices that we use to control sepsis. So it's a method to control antisepsis. So you've heard the term an antiseptic, right? These are chemical agents that you can actually use in the body. For example, Listerine is an antiseptic, right? Iodine, hydrogen peroxide, things like that that you would put on a wound are antiseptics. So an example is using an iodine compound to put on the skin before you make an incision for surgery. So they will always take a swab and put iodine on it. That's what that brown stain is on the skin after you, or when you get a surgery. Or swabbing, an open root canal or a cut with hydrogen peroxide, which I should mention, by the way, since this is a medical class, that is not necessarily a good idea. I know that a lot of people think that you are supposed to, disinfect a cut, if you get cut, with either hydrogen peroxide or alcohol. Now, most people don't use alcohol because it stings like crazy. So most people go to hydrogen peroxide and they like to see it bubble up because they really know it's doing something, right? But there is some fairly solid research indicating that that's actually not a good idea. Here's the reason why. Yes, it does kill the bacteria, but the problem is it creates so much damage to the living tissue that what you end up with is actually a worse healing process and a higher potential for infection because you create so much more damage than was already there. So what is generally recommended, and I suppose it might depend. I mean, if you get a cut, while you're wallowing around in a manure pile, maybe use hydrogen peroxide. But if you are, I don't know, you cut yourself on a branch or something, what they recommend generally instead of that is just in what they call irrigation with saline solution. And the reason why saline solution is because it washes away a fair number of the bacteria, yet it does not cause any damage because it's an isotonic solution, so it doesn't create any damage to the living tissue. And so it doesn't do anything to the pathogens aside from just physically wash them away. But what we rely on at that point is our body's own immune system to take care of the bacteria. And like I said, the studies have shown that the healing time and likelihood of creating an infection are actually reduced using saline solution rather than hydrogen peroxide or alcohol or something like that. And the last example would just be an ordinary hand washing, just washing your hands with, and not even necessarily germicidal soap, just soap is an example of an antiseptic technique, right? So another term we need to define is this term stasis or static. Those are the same root words, and it means to stand still. Stagnant is also from this same root word, so you know a stagnant pond is a pond that's not flowing, right? So that might help you remember that. So a bacteriostatic is a chemical agent that prevents the growth of bacteria on something, either tissues or some sort of an object, right, like a countertop. So a fungistatic, and let me just clarify. This prevents the growth. This prevents the bacteria from continuing to grow or multiply. This does not necessarily kill the bacteria. And so the same goes for a fungistatic. It inhibits the fungal growth. So these antiseptics and drugs often have microbistatic effects because if we try to control them with anything more aggressive, it actually creates toxicity in the human. Okay, so they're not as harmful to the microbes as sides, which we're going to get to in just a second, but they are also not as harmful to the patient as well. So these cidal agents kill. That's what side means. I don't know if we define that or we're going to in a minute, but side means to kill. They don't necessarily result in sterilization, but they can, depending on how they're used. So things we need to consider when we're choosing a method. Does the application require sterilization, or is disinfection adequate? Do we need to sterilize bed sheets? Do we need to sterilize kitchen utensils that we're going to feed a patient? No, because the likelihood of an infection coming from that is not great. We have other methods to be able to control that built into us, right? Innate methods. Is the item going to be reused, or is it going to be thrown away afterwards? If it's going to be reused, can it withstand heat or pressure or radiation or chemicals? Because that will help us decide which method we're going to use. And of course, is the control method appropriate? And those are the things above that we really consider when we're talking about that. And for example, if we use a chemical, is it going to leave some sort of a residue that's going to be harmful? Will the agent penetrate to the necessary extent? Depending on whether it is, on a wound or on a countertop that might be semi-porous, things like that. And is it cost effective, labor effective, and is it safe? Some of these things do a very, very good job, yet they're so expensive that it's just not practical. So what exactly is microbial death? We talk about killing these things, but stop and think about this for just a second. What do we mean by killing something? When we kill a microbe, how do we exactly measure that? So I'm just going to give you a second to think about that. So let's define death. So death is the permanent termination of an organism's vital processes. Here's the problem. Microbes don't really have any obvious vital signs, right? So it's difficult to tell whether a microbe is actually dead. It's easy to tell on a human. We have what we call vital signs: blood pressure, pulse, respiration. If any one of those things is missing, the human is not alive, or at least is terminal, right? On their way to death, either way. But microbes don't have those same sorts of vital signs. So how do we tell if one of them's dead? Well, it's defined as the permanent loss of reproductive capability. In other words, they can't reproduce, even under optimum growth conditions. Because you might say, well, you could, reduce the temperature to freezing, and almost all microbes won't reproduce, right? Well, but that's not optimum growth conditions, because you can actually then thaw the thing back out and they will begin to grow. So that is not defined as being dead, okay? Death is when they're at optimal and they still won't continue to reproduce. There are several things that contribute to the death rate of microbes. So the whole population doesn't die instantaneously, unless it's an atomic bomb or something like that. But we generally don't use bombs to sterilize things. So death begins when a certain threshold of the actual agent is met. So you can't just put necessarily one drop of, some sort of a microbicidal and start killing things, okay? It takes a certain amount. When death begins, it continues on a logarithmic curve, sort of like the population, only backwards when we talked about population growth. So it rises, the death rate gets higher and higher as time continues or as we increase the concentration of the microbicidal. And the reason why it's a logarithmic curve is because certain cells die at a different rate than other cells. So active cells, cells that are dividing, for example, tend to die very, very quickly. Vegetative cells die more quickly than endospores and so on. But eventually, a point is reached at which the survival of the cells is pretty much not going to happen. And this is what we would call sterilization. Another factor is the number of microbes. The more microbes, the longer it takes to destroy them. Another is the nature of the microorganisms in the population. So, as I mentioned earlier, the target population is usually a mixture of bacteria and fungi, spores, viruses, and so they're not all going to die at the same rate. Temperature and pH of the environment is also another factor. The higher the temperature, generally the easier it is to kill, because metabolic processes all occur at a higher rate in general, to a point, obviously. The concentration, which is also the dose or the intensity of the agent, whatever agent we're using. And interestingly enough, higher concentration does not always equal better killing or higher sterilization rate or anything like that. For example, the alcohol that you put on your hands, in the hand sanitizer with like Purell or whatever, there is actually a point where that becomes less effective. So the optimum is like 60 to 80%. Anything above 80% is actually considered is actually less effective. And the reason why that is because you need a certain amount of water in order to denature the proteins, because the chemical reactions that need to occur don't occur in the absence of water. On another side note, since we're on this subject, most people don't use this alcohol-based hand sanitizer correctly. You essentially need to drench your hands in the stuff and then let it evaporate off. Make sure it gets in between, your fingers, all around your fingernails. So I haven't yelled at my wife. I've very politely mentioned to my lovely wife that putting a dime-sized little drop in the palm of your hand and wiping it around is essentially useless. Okay, so make sure you're using a lot of it. So those tiny little things like my wife puts in her purse, those should pretty much be gone after 4 or 5 uses. So anyway, be liberal with it. Your hands need to be soaked. Okay, back on subject here. Ultraviolet radiation is an example of something that could be used to kill microbes. And it happens to be the dose or the intensity of it happens to be most effective at 260 nanometers. So ultraviolet light comes from the sun, right? But there are all kinds of different wavelengths of ultraviolet light. There's UVB, UVA. I don't exactly know what all those are, but it's 260 that is most effective. Most disinfectants are active, are more active or more useful at higher concentrations, but like I said, there's a point, and not all of them are. Some of them are as low as 2 or 3%. Another factor affecting the death rate is the mode of action. So how does it actually kill or inhibit the microorganism? And so when deciding what to use, it's important to take all of that into consideration. The presence of solvents that might interfere with the action, or organic matter that might interfere, or other inhibitors, such as bodily fluids like saliva or blood or feces. All of those things can make it more difficult for the microbicidal agent to actually get to the actual microbe. So these antimicrobial agents have all kinds of ways that they kill things. They target all kinds of different areas, like cell wall or cell membrane or protein synthesis or whatever. The least selective agents are effective against a wide range of microbes. So they target processes or structures that are shared by a wide range of microbes. Therefore, they kill a wide range of microbes. For example, heat and radiation. Now, you may also probably recognize that a lot of these that have the widest range are also more likely to harm a human, and that is definitely the case. And that's more important when we talk about medications and things like that. Selective agents, then, of course, only target very specific cellular components. Drugs, for example, are generally much more selective because we need to be able to harm the pathogen without harming the human, and so we need to target things that aren't shared by the human and the pathogen. So what are some of the cellular targets that are targeted by physical or chemical agents? Sometimes it targets the cell wall. Sometimes it targets the cell membrane. Sometimes it'll target a process inside of the cell, for example, the synthesis of proteins. Sometimes it'll actually target the protein itself. For example, if it's an enzyme, so the protein's already made, it doesn't target the synthesis of the protein, it targets the protein after it's already been synthesized and denatures it. So if the enzymes don't work, then the organism can't continue its processes, and then it's dead, right? So this right here just gives some examples in a table form of things that might happen. So in targeting the cell wall, for example, some of the things that target cell walls are different chemicals and detergents and alcohols. And so ways that they can target is either by blocking the synthesis of it, so it never gets made in the first place, or it actually digests the cell wall. Alcohols are an example of that. Some target the cell membrane. Detergents are an example of that, and that's what this is showing over here. These surfactant molecules are these detergents, and what they do is they go in here and they disrupt the bonding between the lipids, and then it creates holes essentially in the cell membrane, and it basically just falls apart. Detergents are what we use in washing our dishes. It creates a separation of the fat molecules, and that's why you can wash off fats and oils off of your hands. Some agents disrupt this synthesis inside the cell. Formaldehyde, radiation, ethylene oxide, these things all target things like protein synthesis in the ribosomes. Some of them can actually cause changes in the genetic codes too and cause mutations. And so if we create enough mutation, it's going to create enough damage to the DNA or potentially RNA that it can no longer, the information is too messed up for it to continue to survive. Heat, alcohol, phenolics, all of these things disrupt proteins and denature them. Some of these things actually can bind to the active site of enzymes and not allow the other substrates to interact with it, so it plugs it up. We talked about that in a previous chapter. The use of iodine compounds to prepare the skin for surgery is known as disinfection, antisepsis, sterilization, sanitation, or degermation. And as you know, the answer is antisepsis. This is an antiseptic. If you didn't already realize this, it would be very important to know the difference between all of these for the test. Okay, let's put this stuff to work. So how are different ways that we control things with physical control? So we have physical and chemical, right? So we're going to discuss physical right now. So the first one we're going to talk about is heat. So in general, raising the temperature to a certain point is microbicidal, okay? Meaning it kills the microbe. And lowering temperatures in general are microbistatic, meaning it stops the growth but does not kill the microbe. So we're going to start with talking about moist heat. And moist heat is heat with water involved. So either hot water, boiling water, or steam. All of those are considered to be moist heat. And the ranges that we use for this is between 60 Celsius and 135 Celsius. And remember, water boils at 100 Celsius. So we're talking below boiling to above boiling. Dry heat is another method. That's defined as hot air or just an open flame. So we will sterilize, I don't know, like inoculating loops, for example, when we are inoculating bacteria so we don't get contamination. We'll just stick it in an open flame till it gets red hot, and nothing can survive that. So we're talking about 160 Celsius to up to thousands of degrees Celsius. So we'll talk about some ovens coming up in this chapter, and these things get several thousand degrees. So the mode of action and relative effectiveness of heat. It's not all the same. Moist heat and dry heat don't act the same. Moist heat can kill things at a much lower temperature with much shorter exposure times than dry heat, to a point. I mean, if we're talking about thousands of degrees, then that's a different story. But not everything can survive thousands of degrees. So if it's, for example, like a heat-resistant plastic, you can't throw that in a 4,000 degree oven. Therefore, disinfecting or sterilizing that would be more effective with moist heat than dry heat. And you could do it at lower temperatures with shorter exposure times. I guess that was the point I was trying to make. So how this heat actually causes death is because of coagulation and denaturation of the proteins. So it causes them to clump, or it causes them to change shape enough that they are no longer functional. Dry heat dehydrates the cell and removes the water necessary for metabolic reactions, and it denatures proteins. However, it increases the stability of some proteins, therefore it requires a very high temperature to function. At very high temperatures, it oxidizes the cells and can even just burn them completely up. Now, it's important to understand that dry heat can actually make cells more resistant or more able to live to a degree. So this is just a comparison chart showing the difference between moist and dry heat. Okay, so at 121 degrees, you can sterilize something in 15 minutes if you're using moist heat. But if you're using dry heat, it takes 600 minutes. So some of these things are very heat resistant, right? Spores, bacterial endospores, are the things that are most resistant. So we have to actually raise the temperatures above boiling to kill these endospores. And the resistance of them varies among species too, because some of them can create a more durable endospore than others. Vegetative cells also vary in their sensitivity to heat, and we have to heat them up to 50 Celsius for 3 minutes, or ranging up to 60 Celsius for 60 minutes, just depending on which species we're trying to kill. It's important also to understand that the vegetative cells of spore-forming bacteria are just as susceptible to heat as the vegetative cells of non-spore formers. So when we talk about a spore-forming bacterium, the only differentiation is the actual endospore, not the vegetative part of it. So pathogens also, another thing that's interesting, or just something that you may not have thought about, is that pathogens have the same susceptibility to heat as non-pathogens. So I had mentioned that not all microbes are pathogenic. Some of them actually are very beneficial to us. However, when we kill the pathogens, we're killing the non-pathogens as well. Most of the time, this makes no difference if we're killing or if we're disinfecting an instrument, for example. Who cares if we kill the good bacteria? It doesn't make a difference. Fungi, protozoa, and worms are also similar in their sensitivity to heat. In other words, if a certain heat kills a bacterium, it's also going to kill any of those other things as well. Viruses are more resistant to heat because of the decreased structures that they have in them. Tolerance extends from 55 Celsius for 2 to 5 minutes for some, but as much as 60 Celsius for 600 minutes for the more resistant ones. So we have these measurements in trying to determine the death of these microbes. The first one is called the thermal death time, or the TDT. And this is defined as the shortest length of time required to kill all microbes at some specific temperature. The thermal death point, or the TDP, is the lowest temperature required to kill all microbes in 10 minutes. So think about that for a second. The first one is just measuring how long it takes to kill something at a temperature, say, for example, 100 Celsius. So for E. coli, it might be 2 minutes, and for something like Bacillus anthracis, it might be, for the endospores, 45 minutes. I don't know. I'm just making these numbers up. And then the thermal death point is saying, okay, we're going to use 10 minutes as our time frame, and so now we need to figure out what temperature it takes to kill it. So for example, E. coli, it might take 90, well, that wouldn't work for sure. Let's say 100 and, no, yeah, it could be. So let's say 90 Celsius. I don't know, I'm just making these numbers up. But for Bacillus anthracis, it might take 150, for example. Anyway, when considering heat treatment of perishable things like food, the idea is to figure out what temperature we can process this at without and still kill everything in there, or at least enough of it, so that it doesn't cause disease or spoilage, but also not ruin the product or make it so expensive that it's not cost effective anymore. So let's talk about some heat methods, and this is all in Table 9.5 in your book. The first one is boiling water, and this is considered to be disinfection. Okay, just boiling something once, and it's also important to notice or to know when you boil water, it only gets to 100 degrees Celsius. It doesn't go above it. Okay, if you need to get it above 100, you actually have to add pressure. So boiling water, if you stick a thermometer in it, no matter how high your stove is, it will be 100 degrees. It's just like ice water will be at 0 degrees Celsius until all of the ice melts. So I started to say, boiling something one time at 100 degrees Celsius won't kill everything. And the reason why is because there are either resistant cells or spores, and the spores will just remain spores. So if you boil something, you kill the vegetative cells, most of them at least, but you don't necessarily kill the spores. So you may need to wait and let the spores germinate and then boil it again. And you might have to do this several times, this cycling. And remember, there are also bacteria that live above boiling. Now, of course, most of those aren't going to be pathogenic bacteria, but point is, there are still some of them. So generally, 30 minutes is what it takes to kill most non-spore forming pathogens, including tubercle bacillus and staphylococci, which are very resistant. And this is useful in, like, if you have a contaminated water source, for example, you get like flooding, and flood water spills over into the ground or the drinking water system. Cities will put out a boiling alert and what that's to do is to boil the water to kill any pathogens. Or you've probably heard, I remember a raccoon crawled into a water tank or something in not far from where I live and died. And so everybody was getting sick because this dead raccoon was contaminating the water. So for a long time they had to boil their water. It's also useful for disinfecting baby bottles and things like that. Like the nipples of a baby bottle don't necessarily get clean in the dishwasher, for example. So they recommend boiling certain things like that because that way it gets in all the little nooks and crannies and crevices. Food and utensils, bedding, if somebody's sick or something, even just washing bedding in hot water is more useful than like cold, for example. Clothing from the sick room, and so on and so forth. I mean, this list could go on and on and on. Another method is pasteurization. So you've heard of this because our milk is pasteurized. They call it heat pasteurization. Technically, pasteurization is always heat, I think. But there are other methods that they use to kill microbes in things that we drink. Technically, they're not pasteurization, but I think sometimes they still use that word. So what is it? So we apply heat to some sort of a liquid to kill whatever is going to cause the infection or the spoilage. It's showing a brewery right there, and it's also showing beers right here. One thing that maybe I should mention, these things are pasteurized just naturally because they're heated up just in the natural process of creating them. Some are not heated up in the natural process of creating them. For example, milk, it's heated up on purpose just to kill the pathogens. Anyway, the other thing I was going to mention is alcohol is also in these beers, and the alcohol also kills microbes. I think I mentioned that this is back in Britain, a few hundred years ago. They used to all they drank was beer, pretty much, because they realized that when you drank beer, you didn't get sick, but when you drank the water, you did get sick. They didn't really know why. They just knew it wasn't. But what it was is that the alcohol in the beer, and just like I said, the processing, um, keeps the beer from growing microbes that are pathogenic. Anyway, so pasteurization, the goal is to heat up these products without damaging the food. Um, you can heat beer up pretty high, but you can't heat milk up very high, for example, or it starts to damage the actual milk. So they use these heat exchangers that, well, they just heat the liquid up, essentially. So there's two ways. There's the flash method, which is 71.6 degrees Celsius for 15 seconds. And this is less likely to change the flavor because it doesn't damage the milk or whatever it is. And it also doesn't denature the proteins as easily, and so you don't lose nutrients in it. And it's more effective for certain pathogens like Coxiella and Mycobacterium. And the other method is what's called the batch method. And the batch method is heating up 63 to 66 Celsius for 30 minutes. So it's a lower temperature, but for a much, much longer time. And things they will consider is cost and things like that when deciding which method to use. So this pasteurization inactivates most viruses and destroys the vegetative cells of 97 to 99% of the bacteria and fungi. It doesn't kill endospores or very heat-resistant microbes. So as I've mentioned, these are mostly non-pathogenic, so it doesn't necessarily make you sick, but it still could cause spoilage. All right, so there's another thing called Tyndallization. Tyndallization is also known as intermittent sterilization, and it uses non-pressurized steam. Steam is created with pressure, but this is the steam that's leaving whatever the pressurized vessel is. So it's used for things that can't withstand the high temperatures that otherwise would be needed to kill whatever microbes are living on them. So in Tyndallization, whatever you're trying to sterilize is put inside of a chamber that has boiling water in it. So it essentially just creates an environment that's full of steam that you leave it in for half an hour to an hour. So this isn't going to kill the spores. It doesn't get hot enough, and so these spores can withstand this temperature. The other thing is it won't kill the spores in one single use. It's kind of like what I was talking about earlier with cycling it. And so in order to do that, you have to wait for the spores to germinate and then cycle it and kill them again. So these cycles are 23, 24 hours, something like that. So what happens is you kill all the vegetative cells, the spores are still left over, so you give it another day and let them regerminate, and then you kill the vegetative cells again, and you repeat this for 3 days in a row. So this is over here showing, Monday, Tuesday, Wednesday, so 3 days. So the temperatures don't ever get above 100 because that's what steam is made at boiling. If you pressurize it inside of something, you can create steam. But what we're talking about is just having the boiling water inside of the same vessel that's non-pressurized. And it should be noted this isn't completely sterilizing because some really resistant spores can live for more than the 3 days that we Tyndallize it for. So things we use it for are heat-sensitive culture media, such as serum. We use egg sometimes for culture media. Certain carbohydrates, which would break down at higher temperatures, sugars, some canned foods, and so on. So not effective as sterilizing, even though it's called intermittent sterilization. It's technically not. So when you're sterilizing things like instruments and dressings, you actually have to do something different. So this is only a disinfection. Another method for moist heat is the autoclave. So this is steam with pressure, and this is sterilization. This will kill everything. So at sea level, normal atmospheric pressure is 15 psi, or pounds per square inch, which is also defined as 1 atmosphere. And at sea level, water boils at 100. So I had mentioned earlier that water boils at 100 degrees Celsius. What I didn't mention was that at higher elevations, like in Utah where I live, the water boils actually at less than 100. So it doesn't even get to 100 degrees. And that's why you're supposed to boil things longer. Cooking times, you may realize, are longer at higher elevations. If you're into baking and things like that, you probably know this. Than they are at lower pressures, because the higher the pressure, the higher the temperature water boils at. So as I said, steam will stay at 100 degrees Celsius the whole entire time. So it's too low to kill all microbes, and therefore to raise the temperature we have to add pressure. And that's where the autoclave comes in. And that's what this is showing a picture of here. So this is an autoclave. Now this thing looks pretty big here. Some autoclaves are very, very small. So just to give you some numbers, at 20 psi, so 5 psi above the normal 15 psi or 1 atmosphere, the temperature of the steam is actually 109 degrees Celsius, which means that water boils at 109. At 10 psi above normal, the steam's temperature is 115 degrees. At 15, which is double the sea level psi, so 2 atmospheres instead of 1, water will boil at 121 degrees Celsius. And so steam is created at 121. So it's not the pressure that's killing the microbes, but it's actually the temperature that the steam is created at. It's just higher temperature. So we use an autoclave for anything that can handle the kind of temperatures that it takes. So things like glassware, surgical dressings, metallic instruments, liquids that can handle that temperature, paper, certain media, heat-resistant plastics, and just anything that you can put on that list that will handle that kind of heat. It's not effective for things that repel moisture, like oils and waxes, or for things that are harmed by moisture, like powders. You couldn't put baby powder in and sterilize it because you wouldn't end up with powder, you'd end up with a brick, right? So that was moist heat methods, and the rest of what we're going to talk about here is dry heat methods. So obviously no water involved. So the first one is called incineration. And this is where we use a Bunsen burner or just anything that has like an open flame. Apparently that instrument there to the right must have an open flame in it. But when a Bunsen burner, if you probably have used these in chemistry class or something, it's just that little cylinder thing that has a flame coming out the top of it. Okay, so anyway, it just burns things. It incinerates them. Okay, so Bunsen burners, the open flame at the hottest point is 1870 degrees Celsius, and that is going to burn anything that's alive. So things like the inoculating loops that I had mentioned earlier, that you inoculate culture media with to be able to grow a microbe or a colony or whatever. Incinerators are another form of incineration, and that's what that's showing over there to the right. Those things will create a flame inside of them 800 to 6500 degrees Celsius. And so you can use incineration to sterilize things like syringes, needles, all of those things listed in that list there. Animal carcasses is kind of interesting. But when you have an animal that you're doing some sort of a study on, for example, like the CDC may be using an animal to study some sort of a super pathogenic virus or bacterium or something like that. And when they're finished with that, they can't just go throw it in the dumpster, right? So they have to burn that thing to make sure that none of the pathogens survive. So another one is the hot air oven, and that's what that's showing over there on the right. So it's a dry oven, no water, and sterilization requires 150 to 180 Celsius for 2 to 4 hours. So remember, dry heat methods take much longer than moist heat methods. So this ensures that the entire object gets heated, and so if there's endospores down in the deep cracks and crevices or inside of it, it will kill everything. Obviously, these methods require things that are quite heat resistant, right? So we've been talking about heat. How about cold, though? So cold slows the growth of things. That's why it's a microbistatic in most cases and not microbicidal in most cases. For example, in our food, that's why we put things in the refrigerator. It slows the activities of most microbes. So most microbes are not adversely affected by gradual cooling, long-term refrigeration, or deep freezing. Some of them are killed, but most of them will survive, and when they thaw back out, they just continue life as normal. Okay, so temperatures ranging from -70 to -135, that's below zero, right, can preserve cultures of bacteria, viruses, and fungi for long periods. So yeah, not only do they not harm them, this is how we can store them for long periods. So there are actually refrigerators that store these cultures so that they can be studied or used later on. And don't forget, psychrophiles actually grow at freezing temperatures. They can even secrete toxic products. So pathogens that are able to survive several months in the refrigerator are things like Staphylococcus aureus and Clostridium, Streptococcus, so Salmonella. These things are what cause what we call food poisoning, right? And some of them can even cause death. Like botulism, for example. Yeasts, molds, and viruses can all live in refrigeration. Desiccation is a fancy word that just means drying out. Okay, so the vegetative cells that are exposed just to normal room temperatures will eventually dry out. Now, not all of them die. In fact, a lot of them don't die. Some die quickly, though, like Streptococcus pneumoniae, the spirochete that causes syphilis, Neisseria gonorrhea, will die after just a few hours of being exposed to the air. Endospores of Bacillus and Clostridium, however, can live for thousands of years if they're dried out. And some of these bacteria that are actually in like spit, sputum, air, dust, can remain viable even in air, at least for a while. So many viruses and fungi can withstand really long periods of desiccation, like endospores of bacteria. So there's this term, lyophilization. And this is essentially freeze drying. So like if you ever eaten freeze dried food, like those little, I don't know, apple pieces that you can feed toddlers, they just sort of melt. Stuff like that is freeze dried food. Well, lyophilization is freezing and drying bacteria, not to kill them, but to actually preserve them. So the way it works is they're instantaneously frozen and simultaneously the water is vacuumed out. And what this does is, you probably remember me talking in a previous chapter about freezing living things. And the reason why it's harmful is because ice crystals form, and those ice crystals act like knives and just cut everything apart. So by freeze drying, and you can do this with very small things, by freeze drying, it keeps the formation of the ice crystals from happening, and therefore does not cause the damage to them. And so then later on, they can just be rehydrated and they will live. So, it is important to understand not all of the cells survive, but enough of them do survive that you can regrow it later on. So we've talked about temperature, both hot and cold, dry and moist heat, and drying out. Now we're going to move to radiation. So radiation is defined as energy emitted from atomic activities and dispersed at high velocity through matter or space. And we get gamma rays from space, for example. Also, like other things that come from space are like alpha and beta particles and things like that. X-rays, ultraviolet radiation, UV radiation. We actually had mentioned this a little bit earlier. So ionizing radiation is one of these types, and these are gamma and X-rays. Those are both ionizing radiation. And the reason why they're called ionizing radiation is because they emit rays that ionize. In other words, they knock electrons off of atoms and create ions out of them. So we can make these things. Now, these are naturally emitted from space, from like the sun and other stars and things like that. But we can also make them, artificially, I guess you could say. These gamma ray machines have radioactive cobalt in them, and they create gamma rays. X-ray machines, obviously, what you go to the dentist or doctor and get an X-ray from, they create X-rays. Now, when we're using these to like sterilize things or sanitize or whatever the case may be, we use a high level of radiation, much higher than you get from the doctor's office, because we're trying to kill things, right? There are cathode ray machines also that can be used to create radiation. So the dosage of radiation we measure in these things called grays. We used to use rads. But for some reason, they've replaced rads with grays, and I don't really know why. You still hear people talking rads, though, so I'm not 100% sure. Obviously, it's not completely replaced. So exposure in order to kill an organism ranges from 5 to 50 kilograys. So, 50 grays times 1,000, in other words. So all ionizing radiation can penetrate liquids and most solid materials. Things that it can't penetrate are like lead and gold because they're so dense they actually absorb it. Okay, gamma rays are the most penetrating. X-rays are somewhere in the middle. Cathode rays are the least radiating as far as ionizing radiation goes. Now, other forms of radiation like UV radiation is not ionizing radiation, but it's even less penetrating. So irradiating something can lead to a small decrease in thymine, like in food, but it's fairly inconsequential, and they say that just cooking your food basically does the same thing or even more. So another thing to consider is it does produce these short-lived free radicals, which oxidize things and create damage, but they disappear almost immediately because they get reduced by gaining an electron or combining them with something. Okay, certain foods, by the way, don't irradiate well, like egg whites. They get milky. Grapefruit gets mushy. Alfalfa seeds won't germinate after they're irradiated, and there's probably a huge list of other things too. Also, something that people don't understand, because they use radiation to kill bacteria in things like beef, and people think that it might make your food radioactive and not safe to eat, and that's just not true. As far as we know, there's no ill effects of eating irradiated food. So things that we use ionizing radiation to sterilize are things like drugs, vaccines, medical instruments, especially plastics, because they can't handle heat of an autoclave, right? Syringes, surgical gloves, because they would melt. Syringes would melt in the heat. We would need to sterilize them, and so on. Also, we can use it for tissues when we're grafting like bone into somebody. You don't want to put unsterile bone grafts into somebody because you could obviously kill them, or heart valves, even artificial heart valves. So they'll irradiate them and kill the living organisms on them. Okay, non-ionizing radiation is UV light. It's, as I said, not as penetrating as ionizing radiation. A tool that is used for destroying fungal cells, spores, vegetative cells of bacteria, doesn't do anything to endospores, protozoa, viruses, things like that. So bacterial spores are about 10 times more resistant to radiation than vegetative cells. So they can be killed, but you have to increase the time of exposure to a point that it's just not really even worth doing if there's other methods that are better. So usually directed as a disinfectant because it's very difficult to sterilize using radiation. But we use things like germicidal lamps. Matter of fact, I'm sitting here at my kitchen table and I'm looking at a germicidal lamp we have plugged into our wall right now. They even put these in your furnace. You've probably seen this advertised or heard it on the radio or something, where they'll put these UV lights in your furnace, so as the air blows through, it kills living things before it spews them all over into your house. So it can kill up to 99% of airborne pathogens, or for that matter, anything living, microorganisms. So they'll use these lamps in hospital rooms, operating rooms, schools, food prep areas. If you look up on the wall, you'll see these. They're like a bluish colored light, and they always have them covered so you can't look directly at them because it would cause damage to you. This disinfection of air using ultraviolet light is really effective in preventing post-operative infection. So when somebody gets an operation, it helps prevent the infection of respiratory droplets. So if someone's coughing or sneezing or something like that, and also slows down the spreading of airborne pathogens in food processing. So this seems really out of place. I should have shown you this earlier, but I didn't realize this little picture existed because we talked about thymine dimers in chapter 8. But this right here shows what a thymine dimer actually is. So ultraviolet radiation commonly causes these thymine dimers. So from chapter 8, and again, if we haven't gone through chapter 8 yet, if we're doing this out of order, because I think I'm actually going to put chapter 9 ahead of chapter 8, then just listen up here, and when you get to chapter 8, this will make some sense. But A always goes with T, which is adenine and thymine. Cytosine always goes with guanine, right? So if we ever happen to have an AA together, that means there's a TT together over here. And ultraviolet light can actually disrupt this bond and actually cause the bond to be created between two thymines because they're next to each other. And so that's what this shows here. And this is an abnormal bonding, and what happens is as DNA polymerase runs across here, it hits this spot and doesn't know what to do. And if this happens to be in a sequence, for example, that tells, a certain type of cell, like for example, a melanocyte, to not continue to divide, then you could end up with a melanoma, right? Or any other kind of like skin cancer, basal cell carcinoma or something like that. So that's how these things can be bad. But I suppose also this is probably a major reason why ultraviolet radiation kills bacteria, because they're easily ultraviolet light easily penetrates a bacterium and can cause these thymine dimers in it as well. Okay, so let's move on to some other methods that we can use, and these are physical methods, so not using any kind of radiation or anything like that, that we can use to disinfect something or even sterilize it. One is filtration. So filtration can remove microbes from air or liquids. We just run it through a filter using pressure, right? And that pressure can be created by gravity. So fluid is strained, and liquid is a fluid, by the way, or excuse me, gases and liquids are both fluids, but fluid is strained through a filter that only has openings large enough for the fluid to pass through. It does not allow the microbes to pass through. So that's what this is showing here. So these blue arrows are the liquid, like say water, for example, which is definitely the most commonly filtered liquid. And these up here are the bacteria. And they're obviously too big to fit through these holes. And so what ends up passing through is water. And when it passes through, it's sterile. And so they're actually sucking it through using a pump here. But even gravity could do this. It's just that it takes forever. So you need a pump to pump it through. So these holes right here are showing the holes in the filter. And then these little things up here are the bacteria that did not make it through. So they use thin membranes made of either cellulose acetate, polycarbonate, or different plastics that they can actually create holes in somehow that they know the size of. So other things we use is charcoal. If you've ever heard of a carbon filter, that's charcoal. They actually use burned organic matter, and what it does, it creates such a massive surface area. Activated charcoal is charcoal that they heat up really high and then expose to water, and that even creates a higher surface area. And what that does is it makes it so the particles get trapped, which would be the pathogens. They get trapped on the surfaces of this charcoal and never make it through to the other side. Diatomaceous earth or unglazed porcelain are also things that they stuff inside these little cartridges. So the pore sizes can be controlled to make it so you can truly sterilize water, because they're small enough that not even viruses or even prions can pass through. So we partially use this to filter things that can't withstand heat. Now obviously water can withstand heat, okay, so that's not necessarily why we would use that with water. It's just usually a lot cheaper to filter water than it is to do other methods, or sometimes it's just not possible to use other methods. But things like serum, which comes out of the blood, right, or other blood products, vaccines, which would be damaged in heat, drugs, IV fluids, enzymes, media that we would culture things on, all of those things can't withstand heat, and so we would filter them to sterilize them. This is also an alternative method for decontaminating things like milk or beer without altering its flavor. Obviously, you couldn't sterilize them this way. If you did, nothing would get through but water, right? I mean, if you tried to sterilize milk, you're going to end up, by filtering it, you won't end up with milk. You'll end up with water, because it's going to filter out all of the fats and things like that. And proteins, right? So what's left when you filter out all the fats and proteins and sugars is water. Anyway, a very important step in water purification. It cannot remove soluble molecules, however, because they're the same size as the water, and so they actually can still get through. So you could end up with the toxic poisons created by bacteria making it through. So that's a problem. An effective means for removing airborne contaminants. So these things called HEPA filters, which stands for high efficiency particulate air filter, are used in hospital rooms and sterile rooms, so that the air that circulates through it is actually sterilized. How about using something called osmotic pressure? So if we have large concentrations or high concentrations of salts and sugars, it creates a hypertonic environment, and this causes plasmolysis, which means that it causes it to explode. So pickling foods, which uses vinegar, smoking foods, drying foods, all of these things have been used to for years and years and years to prevent pathogens. Most of these, by the way, learned by accident. In fact, probably all of them learned by accident. But osmotic pressure does not sterilize, because what happens is the endospores are not damaged, and so an endospore can be produced in these environments, and then when the environment is adequate, the endospore will then germinate again. So we've mentioned this with like salt pork, right? I don't know why that's not listed here, but when you put too much or a lot of salt on meat, it actually kills the vegetative cells. Something similar to this is like if you've ever had ceviche, which they call it cooking, but it's really not cooking. They use citric acid from lemons to kill the pathogens that might be on fish. So you eat raw fish in lemon juice. It's I guess a South American dish. So what physical control methods would be used to sterilize the following items? Inoculating loops in the lab, glassware that have bacteria on them, vaccines, powders, and oils. So if we were going to sterilize an inoculating loop, the best method would be incineration. If we want to sterilize glassware, like a petri dish or something like that, or beaker or something along those lines, we would use an autoclave. To sterilize vaccines, the best method would be ionizing radiation like X-rays or gamma rays. And if we want to sterilize powders and oils, we would need to use a hot air oven because autoclaving those would cause them to clump up into a brick. That's a big list. All right, so chemical agents. So up to this point, we've been talking about physical, right? Well, how about chemicals? So liquids, gases, or even solids. So they range from disinfectants and antiseptics to sterilants and preservatives. So aqueous solutions are when we dissolve these chemicals in water. So water is the solvent. Tinctures are when we dissolve the chemicals in an alcohol or a water alcohol mixture. So basically if we have alcohol in it, it's called a tincture. So what are the things that we're looking for when we're trying to pick up microbicidal? Speed with which they kill even at low concentrations, how well they are dissolved in water or alcohol, and how long they stay stable. If we have a really good microbicidal but only lasts 2 hours, that wouldn't work, right? You'd have to constantly be mixing it up. Broad spectrum action, meaning it kills a whole bunch of different things without being toxic to humans or animals. How well it penetrates inanimate surfaces, such as, I don't know, porous surfaces like a countertop or something like that. Resistance to becoming inactivated by organic matter. That would be important because if you're trying to sterilize something that's living, it's made of organic matter, right? Non-corrosive, non-staining. How well it sanitizes or deodorizes. How affordable it is, how easy it is to get to. And so there's no chemical that is perfect. None of them fulfill all of these requirements. And so we kind of use different things for different scenarios. But glutaraldehyde and hydrogen peroxide are very close to being able to fulfill all of those. So we evaluate germicides on basically how effective they are in killing. Okay, so high-level germicides kill endospores and therefore can be used as sterilants, meaning they sterilize, right? Intermediate level germicides kill fungal spores but not bacterial endospores. They also kill resistant pathogens and viruses. Low-level germicides can only kill the vegetative bacteria and vegetative fungal cells and only some viruses. So, what are some of the factors that affect this? First of all, the nature of whatever we're trying to kill, right? Some are just naturally more resistant than others. The nature of the material being treated. If we're trying to treat a sponge, for example, certain things might work better than other things. Or a porous surface like a granite countertop compared to, say, a non-porous surface, or whether it's living tissue or not. All of those things come into play. The degree of contamination, how much stuff is on it, right? If it's already clean, quote unquote clean, but there may be something living on it. You wouldn't necessarily need to use the same kind of chemicals that you would if it were, covered in fecal matter, for example. Time of exposure. And this is probably referring to how long you can leave the chemical on it. Strength of chemical action of the germicide. So if we're trying to kill endospores, then obviously it might not work to, use plain old hand soap. So how do chemical strengths and the concentrations of these things work? So we talk about these things as being what are called dilutions. And that is when, we dilute something in something else. So usually it's water, but we said it could be alcohol as well. And we're trying to find some specific ratio that works well. So expressed as, some number with a colon and some other number, for example, a 200 to 1 would be 200 parts of one thing compared to 1 part of something else. So if you're making, gravy at home, you go to Sam's Club and buy one of those packets of gravy, it says mix, 1 packet to 4 cups of water. That would be like a 1-to-4 ratio. Not really, but you get the idea, right? Because that's a powder and a solid. Well, let's just say it's liquid gravy and it's concentrated. How about this? How about a can of orange juice? This works. So we have 1 can of orange juice to 4 cans of water. So that's a 4-to-1 ratio. So you'd go 4:1. That was a good analogy. Actually, not an analogy. That was an example. Anyway, sorry. Okay, so another way we express it is parts per million. So this is used for things that are really, 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 really diluted. Okay, like chlorine in a swimming pool, for example. So we express it as ppm, which stands for parts per million. We could also express it as a percentage. So when we add the solute, we can measure it either by weight or by volume and express it as a percentage. For example, usually the over-the-counter alcohol that you buy in the drugstore is like 70%. You can have it as high as like 91, but usually it's 70. And so you'd say it's a 70% isopropyl alcohol solution, which is 70% alcohol, 30% water. Another factor is length of exposure. So most of these compounds have to be exposed to let me say it another way. These microbes have to be exposed to the compound for a fairly, long period of time before they'll actually kill it. I had mentioned earlier in this chapter about using the topical alcohol like Purell, and then you have to use a certain concentration and leave it on your hands for a certain period of time for it to be adequate. Another one is composition of the material being treated. So as I've said, smooth things are easier to penetrate. Well, they don't have to penetrate because they're smooth compared to porous things, right? If they have a lot of organic material like dirt, for example, could definitely be harder to kill things. So you should clean things before you try to sterilize them, right? Like if you've got fecal matter on some sort of an instrument, maybe you do a colonoscopy with or something. You would want to remove the fecal matter off of it before you go throw it in the autoclave, right? So this right here is, I don't want you to memorize this, please, but this is just gives some example of certain agents and how long and what concentrations, and how long it would take to kill certain bacteria. So Mycobacterium tuberculosis, for example, if we use chlorine at 50 parts per million, it's going to take 50 seconds to kill it. That's why they put chlorine in swimming pools, by the way. But anyway, you're welcome to go and look through this on your own and maybe just use this for a reference later on. Okay, so this table in your book, table 9.9, lists a whole bunch of different chemical agents and the kinds of things they target and the different forms that we find them in and how they actually do what they do, and then some ideas of what we use them for, as well as limitations where they're not, used, or where they don't serve a very good purpose, I guess. So let's zoom in on it. I'm not going to talk about everything on here. We're going to talk about some of them, though. So some things, just because I think they're more significant. So halogens, such as chlorine. I mentioned we use chlorine in swimming pools. It's also used in our water sources. And what it does is when it's combined with water, it creates this hypochlorous acid, HOCl, and that denatures enzymes and kills the bacteria. Iodine down here interferes with hydrogen and disulfide bonds. And remember, those are part of how we create the shapes of proteins, and therefore it disrupts the shape of the protein and therefore makes the protein unusable. And if an organism can't use its proteins, then it dies. Hydrogen peroxide, remember, forms free radicals. That's why we have peroxidases that actually convert these things into water and oxygen, right? So the free radicals are these hydroxyls, and they rip apart molecules, and they're very, very, very toxic. Aldehydes, like glutaraldehyde and formaldehyde. You've heard of formaldehyde. They used to embalm people in it. They don't anymore. But you still might use formaldehyde. And the reason why, I mean, if you think about why are we embalming somebody? Well, it's to stop the microorganisms that will decompose the body, right? Because it kills them. You're essentially like sterilizing a person, basically. Anyway, what does it do? So the glutaraldehyde disrupts the enzymes, basically similar to the hydrogen peroxide. And formaldehyde disrupts the nucleic acids and amino acids of proteins, and again ruins either the molecular, the nuclear material, or let me say that a different way, the genetic material, because these don't even have nuclei, right? The genetic material or the amino acids, and makes the proteins non-usable. So going over to this other page, alcohol. So this is interesting. Concentrations of 50% and higher dissolve the membrane of the lipids and disrupt the cell surface. Essentially, it makes it fall apart, right? So lower concentrations are useless, but also if you get too high, it's just as, well, I shouldn't say just as useless, but it's not as useful, right? Detergents. Remember, they disrupt the lipid membranes as well as releasing the surface proteins of the bacteria. And then heavy metals like silver or mercury, and we don't use mercury as a disinfectant, by the way, but we do use silver, bind onto the functional groups of proteins and inactivates them. So it either hooks onto the active site or the regulatory site. And either way makes it so the enzyme is not used, right? So this just shows how a protein might be affected. So this right here, so here's our protein we start with. Yeah, here's the protein we start with right here. And here's the three choices that we could end up with. Complete denaturation, which is where we take this globular protein and we completely unwind it, and we end up with just the primary structure of it. It might these technically, by the way, these are both denaturation. It might partially denature it, which means it changes the shape of it. And so these little dotted lines are supposed to be bonds, like disulfide bonds. But what it does is it changes the shape of it. And so this thing here doesn't do the same thing as this thing here. Okay. So it can't accept whatever the substrate is. And then this one down here is what I just finished with talking about heavy metals, where the heavy metal will bond either to the active site, which is right here, or the regulatory site, which actually this doesn't have a regulatory site. We could pretend this is the regulatory site, though. Either way, it blocks it and doesn't allow the enzyme to do its job. And this right here just shows what a detergent might look like. So a detergent will have a charged head and an uncharged hydrocarbon tail, and if this looks familiar, it's because phospholipids are like this, which aren't detergents. I don't exactly know the difference, but these R groups here means that they can have any different kind of a group right here. Like, I don't know, I could be totally wrong, but phenols and alkyls and ethyls and stuff like that. And this one down here is showing a benzalkonium chloride molecule, which is an ammonium detergent. So other microbial agents that we haven't mentioned. How about dyes? So dyes will work against gram-positive bacteria and fungi. So they sometimes incorporate these into solutions or ointments to treat skin infections. Very limited application because they stain, so people don't want to necessarily use them, and they have a very narrow spectrum of activity. Acids and alkalis. Remember, acid is an acid, and alkali is the opposite. It's a base, right? So either very low or very high pHs can destroy or inhibit microbial cells by just damaging them to the point where they can't function anymore. Problem is, if they damage the microbe, oftentimes they also damage the patient. So a lot of times we can't use these, at least on a person. So ammonium hydroxide is an example that can be used in cleaners, deodorizers, detergents, things like that. I think that's the thing that's in Pine Sol, by the way, which you do not ever want to mix with Clorox because it creates a poisonous gas and it will kill you. Let's see, what was that last one? Organic acids used in food preservation. So this would be like vinegar, acetic acid, right? That's an organic acid. Use it in pickling things. Citric acid. I mentioned ceviche, but you could also use that in some sort of a stored food, I suppose. All right, so here's some common products that you're familiar with: Lysol sanitizing wipes, Clorox disinfecting wipes, and shows what the chemical is in them and what category of antimicrobial they are. So Lysol and Clorox wipes both have this dimethylbenzylammonium chloride, and it's a detergent. Tilex, Lysol mildew remover, these things have sodium hypochlorites, and they're a halogen, and so on. Triclosan, these are in antibacterials, and they're phenolics. So a normal hand soap is not an antibacterial. To be considered an antibacterial, they have to have, I don't know of anything that has something besides triclosan in it, but there probably is other things. Okay, so we're just going to kind of recap what this whole entire chapter was. Now, this actually is in the beginning of the chapter, but it didn't make a lot of sense to me to talk about it at the beginning because you didn't know what any of this stuff was, but now you do. So let's kind of try to put this back all into sort of a nutshell. So physical agents were divided up into heat, which was also divided further into dry heat, which included incineration, which was a type of sterilization, or a dry oven, which was also a sterilization. So besides dry heat, we also had moist heat, which was either pressurized or non-pressurized. So sterilization would be like autoclaving, that would be pressurized. Non-pressurized would be a disinfection. Also under physical, we had radiation, which included ionizing radiation, which was X-ray, cathode, and gamma rays, which were sterilizing, or non-ionizing, which was ultraviolet, which is just a disinfectant. It is not a sterilizing. Chemical agents included either gases, which could be either sterilizing or disinfecting, depending on the gas and what you're trying to disinfect, or liquids. So either on animate objects, living things, would be an antisepsis, so like mouthwash or iodine or alcohol or hydrogen peroxide, those would be antiseptics, right? Or on inanimate objects, which could be a disinfection if you have the right liquid, or would be I said that wrong. Could be disinfection or a sterilization if you have the right liquid, because some of them will sterilize. And then the last thing would just be mechanical removal, which would be filtration. You can either filter air, which is considered to be a decontamination, or you can filter liquids, which, and this says sterilization, but it could also not be sterilization. Not all filtration sterilizes, but it could be. So hopefully that sort of kind of puts everything into perspective on what we just talked about. So which of the following antimicrobial chemicals are considered sterilizing agents? Chlorhexidine, hydrogen peroxide, alcohol, acetic acid, or none? And the answer is hydrogen peroxide. And that's chapter 9.